I'm Seng Chan You from the University College of Medicine from Korea, and please call me Chan. Today, I'm going to talk about how to conduct Odyssey network study, and I'd like to focus on how to develop protocol and why we need to develop protocol and how to set up the reproducible computation environment for the SOS challenge. Since we had the first SOS challenge tutorial yesterday in my time zone, 70 hours ago, which was led by Mark Suchard from UCLA, and I highly recommend to watch Mark's terrific tutorial for the SOS challenge. And since I'm not the good one to reproduce the Mark's presentation today, so I prepared my slides by myself, and I'd like to focus on the why we need to develop a protocol and how to set up the reproducible environment in my way. So since many of the participants of this meeting is not familiar with Odyssey, I believe, so let me remind what Odyssey is and what Odyssey pursues. So Odyssey, the Observational Health Data Sciences in Informatics, is an international collaborative consortium applying open source data analytics solutions based on AMOP CDM to a large network of health databases across the world. Actually, it was really old-fashioned slides. So we do have more than 40 countries as a participants of Odyssey right now. The mission of Odyssey is to improve health by empowering a community to collaboratively generate the evidence that promotes better health decisions and better care. And our vision is a world in which observation research produces a comprehensive understanding of health and disease. And I'd like to remind the objective of Odyssey to you. Innovation, reproducibility, openness, community, collaboration, and beneficence. And I believe that the SOS challenge is really highly compatible with this objective, 6 objectives Odyssey. We pursued innovative and reproducible research through open community and with the collaboration and beneficence. Actually, even for me, it is not easy to conduct Odyssey network study. So I asked ChatGPT about how can I conduct Odyssey network study, and the ChatGPT answers that Odyssey network study involves collaborative research across multiple institutions that use standardized data models and analysis method to answer research questions, and the ChatGPT suggests several steps to conduct Odyssey network study. So you need to join the Odyssey network. You need to define your research question. You need to identify data sources. You need to harmonize data and develop your analysis plan. Then you can conduct your analysis. After conducting your analysis, we need to interpret the results, and then finally we can publish our findings. Overall, conducting the Odyssey network study requires collaboration, coordination, and adherence to standardized data models and analysis methods. So just it's awesome. ChatGPT was awesome. Yeah, I've got the real actual evidence. Yeah, this is really ChatGPT answers to me. So you need to join the Odyssey, and there is an Odyssey homepage, odyssey.org, and in the tab, you can see the New to Odyssey tab, and you need to join the Odyssey Microsoft Teams environment, and you can join the Odyssey forum discussion. So all of our discussions and the materials will be shared through the Odyssey Teams, and we're gonna stick to this Teams channel. And I highly recommend to read the Teams best practices and guidance before using Teams in Odyssey. So this is how our Teams looks like. So if you join the Odyssey team, then you can see the the Odyssey main team. And then there is channel for the SOS challenges, such as anti-VEGF and kidney failure and fluoroquinolone study, or the other two, the PML incidence and exposure to risankizumab. Yeah. And there is a team's best practice and guidance. Please read this. There's already there's several threads in our teams. And after this, you need to define your research question before conducting to conduct your network study. So in our session, we're gonna focus on the fluoroquinolone research question. So together with Jung Kim and then Jack and Nicole from Australia, we suggest the research question that is fluoroquinolone use really associated with the development of aortic aneurysms and aortic dissections? And Jack and Nicole, if you want, I can play the previous YouTube, or you can present your study in this session lively. Which one do you prefer?
Jack, tell us about your study.
Yeah, no, thank you, Chan and Patrick. So yeah, I guess essentially the main thing that we're trying to do is sort of characterize, estimate, and predict the risk of aortic aneurysms and dissections following the prescription of fluoroquinolone antibiotics. So as you've got there on the screen, our primary questions are, does exposure to fluoroquinolones actually increase the risk of experiencing those aortic events within a year after starting the quinolone? And then for patients with selected conditions which quinolones are indicated for, what is the probability that they'll go on to have an aortic aneurysm or dissection following the initiation of a quinolone? So all encompassing.
Yeah, thank you, Jack, for description your study. And you know that there is a YouTube recordings and Jack's. There is a wonderful presentation of Jack, so you can see the whole the more detailed background of this study and what we are gonna do during this SOS challenge.
If I could just add, I'm particularly excited about this question because this is one of the rare instances where observational data has actually been used to make an assertion that a drug exposure is related to an outcome. If you read the product labels for fluoroquinolones, it says epidemiology studies have shown that there's an increased risk of aortic aneurysms. But what they don't say is how reliable is that evidence and whether or not we actually believe it's true. So I'm really excited that Chan and Hongju and Jack and Nicole have brought this question to the table, because I think that from my reading of all of the previous published papers, there are some questionable epidemiologic designs being applied. So I don't believe that the answer is really a firmly settled decision about whether or not there's a true effect. And I think we have a chance to produce a very credible piece of evidence that can really meaningfully inform clinical practice and regulatory policy about these products, because a high quality study that shows that there's an effect will strengthen the evidence of understanding that relationship, and a high quality study that refutes the effect potentially raises questions about the small handful of previously conducted studies with questionable designs that are currently the evidence base to make this assertion on the product label. So I'm really excited about this study because I think we have a really profound opportunity to make a big impact by working together as a community.
Yeah, I agree with Patrick. So I also, all of our team has some question on the reliability of previous studies, and their results are conflicted. So we're gonna show we're gonna stick to the best practice of Odyssey to conduct epidemiological study or prediction study, and we're gonna learn how to conduct this study during 2-month SOS challenge. So why does Odyssey requires to develop analytic plan first before conducting a study? And what do epi studies currently look like? So usually when we have some questions, or even though we do not have any questions, but sometimes we just got some results from professor or just clinics, and then we need to run some studies by using this data set. So by using Excel or SPSS, we are generating some results with estimates and confidence intervals and p-values. When you start the study, usually I've got the clear goals and clear endpoint, but while running the study during on year, there are several challenges I've never expected, and it was hard to track the overall study steps. So most epidemiologists view a study as a journey from data set to paper, and the protocol might be your map, and you will come across obstacles that you will have to overcome. And you had several steps required manual intervention, and in the end, it will be impossible to retrace your exact steps. And unfortunately, to hack our ways to scientific glory, we sometimes hack our p-values. So there is a website you can simulate the p-hacking. So by selecting several variables or deselect several variables, you can modify your p-values for your scientific glory. And this is what many of us are doing until now. So what should Odyssey studies look like? We believe that the Odyssey study should be like a pipeline. So we want to make a fully automated process from database to paper. So in the Odyssey, the performing a study means building the pipeline from the database to the paper, and this pipeline should be reproducible. So there are some tools for reproducible research, and some of these tools are introduced by Mark 7 hours ago, and I'm going to talk about more about Git and Docker. So we need version control and the research. So these are my actual data sets for the study. So in 2014, I've got some mail from Mayo Clinic that they need my data for their meta-analysis. And actually, it was impossible to identify what exact data set I've used for my published paper, because there are so many final raw data. So which raw data is actual data I've used for my study. And because the Mayo Clinic requires my data, it was really impending project for me. And so there will be final revision 2, final revision 6 comments, final revision 8 with comments, corrections. And usually we do not use the real final data set. We usually use them the version 6 rather than version 8 in the final papers. So there are multiple files for one file, and it is difficult to know who changes file, different among versions, and how these are managed by multiple members, and how these versions are updated. And there is no, logistics about this version names, And finally, it is really difficult to manage a history of file. So use GitHub as a study hub has many advantages over this. So Git makes them possible to control the version of study protocol, and it can effectively manage source code. And we can collaborate efficiently with other researchers through GitHub, and we can neatly package the research by to the end-to-end study pipeline. So this is kind of the experience of mine, how to collaborative develop the package or study package using GitHub. You can see that many of members actually work together within Git, and we usually fork or use branches to protect main branch, and the many ideas or many developments actually merged into the main branch, and it was introduced in the Mark's presentation 7 hours ago. So again, it is highly recommended to watch Mark's slides and the YouTube video to before using GitHub in Odyssey. So, and we need to develop end-to-end executable analytic program. And this is an example of my previous study about this. So let me share my previous study repo. So this I've committed more than 100 times to for this one single study, and you can track every history of my commit or improvement of the study package and why I need to revise the package. So when the editors or reviewers of study requires me to add some more result, then I need to, I had to add some introduction, or I had to fix the. I need more means and standardized difference of standard deviation of age, which the reviewer required me. So in this code, you can see how I add this. I can draw this means and standard deviation of ages in the code. So you can easily track your study, and it can easily learn from other studies too. And I usually manage my issues in the study by using issue in the GitHub. So when I thought that I had to add a 28-day blanking period for the outcome. So I considered, and I've actually did this in this. Sometimes I tagged some Git commits how I did do this. I did this. So yeah, in this commit, I added the introduction term analysis for the maintenance of aspirin dosage. So we can track how I add this feature to add introduction term analysis for aspirin maintenance, and you can learn from the others study to learn to add your own functions to your study package. So there are many study packages, and you can see to learn from your coworkers in Odyssey. Still, I think there is a problem in reproducibility. Previously, George asked me that he had difficulties installing the packages in his local machine, and actually, I have some difficulties to install the packages of Odyssey in my local machines, because most of the Odyssey packages are developed by the old guys such as Mark Suchard or Martin Schumi, which prefer Java. So it's really hard to install Java in my Windows machine. So yeah, I do have similar issues, and I believe that some of you have these issues in your local machines, because we're not that old. I'm sorry, Mark and Martin. I don't see them in here, right? Okay. So yeah, there is a
I'm
problem.
wondering what that makes me, Chan. Oh, you're calling Mark and Martine old, and I don't know what that makes me, but I hear you.
Yeah, I didn't realize you. Sorry. Please forget this. Yeah, again, there is a problem in reproducibility, and the Odyssey pursues reproducible science. So it's really issue in Odyssey, and there are several conventions we have. And another convention I'd like to looking for is the Docker. So what is Docker and what for? Docker is an open platform for developers and sysadmins to build, ship, and run distributed applications consisting of Docker Engine, a portable lightweight runtime and packaging tool, and Docker Hub, a cloud service for sharing applications and automating workflows. Docker enables apps to be quickly assembled from components and eliminates the friction from between development, QA, and production environments. Okay, seriously, what is Docker? Docker is a very lightweight abstraction using recent Linux kernel features, which lets us to run code in cheap and easy units, which is container. We can share containers across OSs, across times, which guarantee the reproducibility of the study. So by capturing the computational environment, Docker can enhance the reproducible environment for this, because there is a substantial challenge in reproducing analysis in installing and configuring the web of dependencies of specific versions of various analytic tools. And as you may know that Odyssey tools are really has really complicated dependencies for to build. So by capturing the computational environment in Docker, we can enhance the reproducibility. So, and I've recently developed the Docker images compatible with the recent Atlas version 2.12. So you can use this Docker image to set the reproducible environment in your local machine. And if you have any troubles, please let me know. So why we are doing the Save Our Sisyphus challenge? The task of the research idea from design through execution through publication can seem a daunting challenge, much like rolling a boulder up a hill. The task is all the more challenging when researchers try to go it alone, and each step requires a distinct set of skills. Observation study design requires epidemiological understanding and statistical methodological expertise. Implementing a study design requires statistical programming ability. Interpreting and reporting research require domain knowledge of the clinical problem. But when you are a part of the Odyssey community, you never have to go it alone. And as a team effort, what seems an arduous task can become an efficient and effective process. So this is why we are doing the Odyssey Save Our Sisyphus challenge. And during the 2 months, I'd like to conduct a network study, and I'd like to educate how you can conduct the network study by yourselves with the members in the Odyssey community. And I want you to learn how to work work together in this community with the open mind. So this is the end of my slides. If you have any questions about the study question about quinolone or my presentation, please let me know. Oh, and I didn't know that it is recorded. Sorry, Mark and Martine again.
Thank you, Chan. I'm going to post. I'm going to post in the chat the link for the Save Our Sisyphus Challenge homepage. And Chan mentioned Mark's tutorial that took place. I'm going to post that there as well.
again, since I think that many of the Asian Pacific members are not so familiar with the Odyssey's philosophy or Odyssey study and how it proceeds. So it's really abstract of the study process. And then again, please watch Mark's presentations. She's in the YouTube.
Yeah, and I guess if I could just add Janet, for the benefit of everybody on the call, what we're going to do over the next 2 months, as Jody walked through, we're going to walk through all the steps of doing this study step by step. So, we've started with this really important clinical idea where we don't know whether or not fluoroquinolones increase the risk of aortic aneurysms. And we're going to march through every week for the next 9 weeks. We're going to walk through the steps it takes to try to answer that question. And today, the step was, let's create a network study. Let's initiate a place where we can collaborate. Let's produce a place where we can start talking to each other about what is the question and why are we thinking we need to work on it. And let's also open up the very first step of the process, which is we need to create a scientific protocol that states what is it that we're doing that we're going to try to use our data to answer. And only once we have a scientific protocol in place that describes what the question is and how we're going to answer it, should we move on to creating the analysis code to execute the study using some agreed definitions of exposure and indication and outcomes, which should be described in the protocol. And only after we get as many data partners as possible to run the study will we have the results that we're going to interpret together as a community. So I think this week it's most important that we all make sure that we understand that clinical question that Jack started to outline, and that we have a shared understanding of what we're going to do over the next couple of weeks, because we want that plan to be described in the protocol, which is going to be posted on our GitHub repository as a place where we're keeping all of the content that we're using to monitor our collaboration together. So if there's given that there's some time available, it'd probably be helpful if we could have a brief discussion to make sure everybody on the call is really fully aware of what the fluoroquinolone aortic aneurysm question is really all about, and how we imagine going about answering that question that's open to the community at this point.
And Patrick, if we have time after that, can you walk us through really quickly? Because you have 9 weeks, right? I see it on the website, but maybe the folks here don't understand what each week is about. So if we can walk through what each week is about, like next week, it's a tutorial on data diagnostic. Why is that important? Or why is phenotype develop? Well, I think I know what phenotype of development is, but what's phenotype evaluation, right? If we can just walk through a little bit of what each one of these are and what to expect each week, that would be great.
Yeah, so maybe Jody, do you want to go back to your slide that showed the roadmap, and maybe we could just talk through that with a little bit more detail, and then maybe we turn it over to Jack and Nicole and Shannon and the team, and then we'll dive into the clinical question.
Can everyone see my screen?
Yep.
Yeah.
We see that.
So data diagnostics. So for next week, data diagnostics tutorial will be led by Claire Blacketer. Patrick, do you want to go through each one, or?
Yeah, I'm happy to. So yeah, but let's just go through the list. So
Okay.
database diagnostics. So the idea here is we're starting with an important clinical question, which is, do fluoroquinolones increase the risk of aortic aneurysms? So to answer that question, we need to figure out which databases across the community have the necessary data elements to answer that question. Very simplistically, in this case, it means we need to figure out which databases have fluoroquinolone exposure, which databases have aortic aneurysm diagnoses, which databases have the necessary information to have some comparator group, which is like other types of antibiotics, which databases have the indications associated with fluoroquinolones, and Jack's highlighted that we're going to focus on urinary tract infections and community-acquired pneumonia. And then these outcomes typically are the kinds of thing that we're not expecting to happen on the same day that you take the antibiotic, but they might accrue over some period of time. And specifically in Jack's hypothesis, we'd like to evaluate the risk up to a year. So that means that we're trying to find databases who have diagnoses for the indication of urinary tract infection, diagnoses for the outcome of aortic aneurysm, drug codes for fluoroquinolones and other antibiotics, longitudinality, meaning that you have to have at least a year of follow-up time, and depending on how we define our outcome, we may require other data elements. So for example, in the previously published papers about aortic aneurysm, prior researchers required that to find an aortic aneurysm in your data set, you needed a primary, a diagnosis in a hospital visit within the primary position for which you had a procedure code for either a diagnostic imaging activity or a therapeutic surgical intervention. And so our first step next week will be to assess data diagnostics to basically determine which databases across the Odyssey community have all the necessary data elements to possibly answer the question that Jack and the team have laid out. And some databases will have some but not all of those elements. And if, for example, if we require hospitalization for the aortic aneurysm, then a database that only has general practice diagnoses would not be sufficient. So next week we'll be going through how do we actually execute a database diagnostic analysis by first characterizing individual databases across the broader Odyssey community. We hope that all data partners who are participating in the Odyssey community and want to participate in the SOS challenge studies will run a piece of code called DB profile, which will characterize your data set and allow us to determine whether or not your database is fit for use for any of the 4 questions. But next week on this 7 p.m. Eastern time call, we'll focus on the fluoroquinolone question. From there, once we've identified the subset of databases that are eligible to consider using, we'll go into the phenotype development and evaluation process. So here we're going to go through the specific process of saying, how do we find systemic fluoroquinolone exposure in our data sets? How do we define urinary tract infection and pneumonia? How do we actually find a case of aortic aneurysm in our data set? And so we'll go through the process of creating a phenotype using Atlas. And then once we've created the phenotype, we'll want to run a series of phenotype evaluation tools, including cohort diagnostics, and for those that are interested, also running P-Evaluator as a way for us to quantify measurement error of our phenotypes. Once we have the phenotypes in place, we're going to walk into doing our analysis design. So Jack outlined that we already have a very well-defined characterization question of how often does aortic aneurysm occur within fluoroquinolones. We have a good estimation question, which is, do fluoroquinolones increase the risk of aortic aneurysm more than other antibiotics? And we have a really good prediction question, which is, amongst people who start a fluoroquinolone, who's the patient that's most at risk for having aortic aneurysm? So all 3 of those questions, we will design the analysis so that we can specify all of the input parameters to run that analysis across the network of databases that have passed data diagnostics. Once we have run the analysis, so that's the network execution, we'll then go through a process of evaluating study diagnostics. And on this call, Nicole's going to lead us through that session. But effectively, what we're going to do is we're going to understand which studies seem to have produced reliable evidence on which databases, and which studies may have failed to study diagnostics, meaning there's a reason why we shouldn't trust it. And we'll take a critical look to see what evidence are we left with, meaning we've passed diagnostics. What evidence did we have to decide not to trust because it had some critical flaw that we were able to observe a priori. Once we've got our subset of databases with valid results that have passed diagnostics, then we'll go through a process of evidence synthesis, where we'll execute a meta-analysis across the results to produce our composite answers. And at that point, we will have all the evidence in hand. And our last session will be focused on interpreting the results. So how does the knowledge we gain from our Odyssey network, what have we learned from that, and how does that compare to what we already knew from clinical trials and other observational studies? Throughout the process of doing all of those steps, we're going to be actively managing writing the publication so that we can chronicle what we've learned along the way. So starting today, we can start working on the background section. And in the MS Teams environment, we already have a draft protocol that's been posted. So we can all start to think about exactly how are we going to answer this question. And as we go through the process of phenotype development and analysis design, we can finish up the method section of our paper. And then once we run the results and have findings, we can summarize our results section of our paper. And then at the end, we'll summarize our discussion to really try to contextualize what we've learned and why it matters. So, we're only committing an hour a week on these community calls together to showcase the specific tasks. But really, if you put each of these tasks together and just focus on it, what we're really giving everybody is a step-by-step cookbook of how to run a network study, where at the end of this 9 weeks, it's going to be exciting when Jack has a finished paper that we can submit to a journal. But I also hope that all others on the call are going to be sufficiently inspired to say, that's not that big of a Sisyphusian task, but that you all could feel empowered to run your own network studies and march through the same set of 8 tasks one by one until you're done with your study. But together as a community, if we do these 4 studies, or when we do these 4 studies in the next 9 weeks, it should be a very clear demonstration of how it's possible to take a really important clinical idea and translate that all the way into reliable evidence that we can all trust and feel comfortable sharing with the broader research community.
Thank you, Patrick. Again, we have to write a paper in parallel with the tutorials. So let me share my screen again. So there is Teams channel for the quinolone study, and Jack actually uploaded the protocol and draft of paper already. And please review this protocol and draft, and please contribute to this protocol or draft of paper by reviewing or adding some comments on this paper in the Teams. And so we're gonna stick to the MS Teams to discuss and then develop protocols or papers. Also, I'm gonna create the GitHub repo for this study to collaborate to work for the study package and the protocols in the GitHub too.
I have a question.
Yep.
I'll ask for those who are maybe similar to me, maybe no one's similar to me on this call, okay? I know nothing about the protocol. I know nothing about medicine, but can I still review this? Is it still useful for someone like me to review it? One, so that I can learn an understanding of the background of this, as well as maybe grammar checks. I know, Jack, you're great at grammar. Your grammar's probably better than me, but is that something, you guys are looking for? So I feel that I'm a bit more useful, I guess.
Yeah. First of all, Odyssey is a open community to work collaboratively to generate evidence, and you will be useful, but you don't need to be useful. Again, so first of all, you don't need to be useful. You can learn from our study and the best learning process will be participating on study as a member, participating member or the joining authors of the study. So, and then, yeah, you will be useful if you have any experience in medical research or analytic tool development, or just because and you have some experience in the human society, and so there will be something we can learn from you. Kung Fu in the Chinese philosopher Kung Fu says that if there are 3 people, then there will be one thing we can learn from these 3 people. So
Cool. Thank you. I got another newbie question, okay? Because I'm gonna be a very newbie right now. You
Please.
just showed Odyssey Teams. Is that a channel that I have to get requested to be added to, or how do I find it? Where is it on Odyssey Teams?
So you need to join the Odyssey Teams first. So in the Odyssey homepage, there is a tab for new to Odyssey, and there is an introduction how you can join the Odyssey Teams environment. So please go enter this tabs and to join the Odyssey Teams environment. And then this is a team, the main team of Odyssey. If after you're joining the Odyssey team, then you can see this main team. And this main team, there is a channel for the SOS challenge.
So we're already in Teams, right? Like I'm already in Teams.
Right.
It's just underneath Odyssey. Okay, and I don't have to get access to it. I don't have to go and request access at all.
Yeah, there's no special access, Mui. It's possible that for some on the call, if you are already in the Odyssey Teams environment, you may not see the channels that have been created for the SOS challenge because they may be listed under what says hidden channels. So if you are in the Odyssey MS Teams, you may need to go to hidden channels. And if you just click on the button that says hidden channels, you'll then see all the things that are hidden from you, and probably the 4 studies that are doing here may be listed in that list. I don't remember, Jody, and maybe you probably could change it, Jody, when you set it up. There's an option to set to say, make it shown for everybody. And I'm not sure that you click the make it shown for everybody button. So for those on the line, if you don't see it, go ahead and add it. And then Jody, for you, you can probably change the parameters of it to try to make it be shown for everybody.
That is exactly where it was. I just checked my phone. It was in the hidden channels. So I unhit them for myself.
Okay, great. Yeah, thanks for the tips.
Thanks, Patrick. I'll go ahead and double check that.
So Jack, can you provide a bit more insights on the study itself? Again, this is a non-medical person, so I want to ask a bunch of stupid and dumb questions. I apologize in advance. That's my disclaimer.
We're just going to share our screen so we can show some slides. Sec. The overview.
Sorry. Yeah, I thought you were going to say. So do you want me to run through the presentation a bit again, or just sort of contextualize the questions for you?
Yeah,
I think just the question, given we only have 6 minutes left.
Yeah. So I guess in terms of the background, they're pretty broad spectrum antibiotics, usually used for more severe infections. But yeah, their use is rising internationally as, you know, there's more and more bacteria that antibiotics are resistant to. In the past, we thought they were pretty well tolerated and not too much of an issue, but there have been, you know, some post-marketing and epidemiological studies, as Patrick said, I guess raising the awareness of these risks. But the studies that I guess went into these recommendations being made by the regulatory authorities, we characterize them as being sort of moderate quality.
Can I just interject there? For those of you, Mui, as you said before, some people don't have, you know, a good background and clinical background. It might be worth those of you that are interested to actually have a look at those 4 studies. and have a read of them, and particularly the background, how they approach their study, you know, what study design they use, because actually all 4 of them had totally different study designs and different ways of going about answering the question. And, you know, just reading those 4 studies can really help to understand what we're trying to do and some of that clinical background. So yeah, one of my suggestions is really to have a look at those 4, and it would be really useful. Yes, Millie?
So I will, and that really is great suggestion, right? And while if we have questions, like if I read this over my weekend, if I have questions, can I post those questions on MS Team, or can I ask them at the next weekly call?
Do both, because I'm a bit the same. I have a pharmacist background, but not a pharmacoepi background. So I'm also learning how to interpret the studies as well. Yeah. So that'll be awesome if we can have, more discussion about that for sure.
Cool. Again, I apologize if it's not going to be like the most smartest question in the world.
No, that's excellent. Yeah. So these were the 2 questions that we talked about before. So does exposure to quinolones increase the risk of these aortic events within a year? And then looking at specific indications for quinolones, what's the probability that if people are prescribed these medications for those indications, that they go on to have one of those events? And then here are sort of, I guess, the 3 different parts that we're trying to address through the study. So characterizing the risk, estimating the risk as well, and also predicting the risk. So each of them have some sub-questions, but we can adapt them as needed as well. The rest of the information in here, we've already put into the protocol on the Teams page. The active comparators can change depending on what our data collaborators have.
And just perhaps go back. So this is actually quite an important table, I think, for those of us that are really interested in how we're going to do this study, because we have this situation where we've got fluoroquinolones that can be dispensed in different settings for different types of infection. And, you know, the active comparators may potentially be different because of that, because what we want to understand is what is the impact of the medicine on the risk of the outcome, and what is the impact of the underlying infection, or is the outcome due to the underlying infection, and stratifying or, you know, restricting on the setting, which is kind of like a proxy for the severity, I suppose, of the infection, is going to be really important to unpack the risk. So for those of you that are clinically, you know, inclined, it'd be great for you to review this and have a think about, in practice, is this what happens? Have we got any fluoroquinolones there that are used in other settings and that we don't know about, that kind of thing, and what would be potential active comparators? And we'll do all of this as well through the study, but if the clinical brains can get together and think about the clinical situation, and the, you know, methodological brains get together and think, well, how are we going to approach this given that clinical situation, and how do we you know, get rid of the impact of confounding that's likely to be in this question. So so have a think about this table. I think it's really critical for their implications for the study.
Yeah, so there's definitely not, the table's not a like, it's not rigid, it's definitely very flexible. I think the risk prediction model will be pretty cool to design as well. That's something that's definitely hasn't really been done by any of the other previous studies. And the rest is all just extra information on how you might be able to contribute, which we've already talked about.
Thank you, Jack. Thank you, Nicole. I just want to highlight a question and a tip that's in the chat for those who haven't seen it. So a tip from Amir is that you can pin the channels. So let's say if your channel is hidden, you can pin that within the Odyssey Teams environment to ensure that that shows up. A question that we also have in the chat is, do we have instructions available for data diagnostics? Yes, we do. And in fact, next week tutorial will be exactly on data diagnostics, and that will be recorded. That would be available on the SOS challenge homepage. These recordings will remain up there, so you can go back and watch several times as well. Thank you.
Thanks for joining today's call, and see you later. And do the SOS challenge together. Thanks for all.
Happy reading, everybody. So I hear that now
Thank you.
everybody's going to go read this protocol, right? That's what I just heard. And you're going to all provide comments on the channels or come back next week with questions.
Yeah.
Okay.
Please.
Okay. That's everyone's homework assignment, including myself. So if I can do it, non-geeky, non-medical person, you guys can do it too. Okay.
Yeah. Perfect.
Thanks, Mary.
Thank
Yes.
you.
All right. Bye, everybody.
Thank you, everybody.
Oh,
Thank
there's
you.
there's
There's
a
a
question.
hand up.
There was a question. A hand was up.
I think
Yeah.
I saw your hand up.
Yes. Unmute, please.
No, that was a mistake. That was a mistake.
Okay.
I'm good.
Ah!
Yeah.
We take mistakes too.
Have a good evening. Have a
Wait.
good day. Yeah,
Have a
no,
nice
I'm
one.
leaving. Yeah, bye-bye.
Bye-bye.